This is the recorded lecture for Chapter 6 on the cell. All right, there's the highlighter. Okay, so this is an organism that lives in freshwater environments, for example, like a pond, just a regular pond or a lake, but freshwater environments. The organism's name it's a paramecium, and you can see that it is a single-celled organism. This is just made of one cell, and this is what it looks like under high power, under the light microscope. Well, it's probably not a light microscope. I don't know, really. It's probably an electron microscope because there's so much detail. But anyway, this is a paramecium under a microscope. And you can see on the outside surface of this paramecium are these little hairs. They're called cilia, and they help it to swim. I can tell you for the paramecium that it is a single-celled or unicellular organism, and I can look at this cell and tell that it's a eukaryotic cell, a cell that has a nucleus and membrane-bound organelles, versus a prokaryotic cell. Now, there's three domains of living things. There's the bacteria and the archaea, and they're always unicellular, and their cells are prokaryotic. They're going to be about this big compared to eukaryotic cells. So look at this little yellow dot that I just made. That's about how big, maybe even smaller than that, a bacterium is going to be compared to a protozoan, which is still unicellular, but it's a eukaryotic cell. So eukaryotic cells, the ones that have a nucleus and membrane-bound organelles, like you see in this one, they're going to be larger than prokaryotic cells. And prokaryotic cells will be smaller, and they won't have a nucleus. So we're looking at a paramecium, which is unicellular, but it's a eukaryotic cell. Okay, so here is a picture of a typical, looks like a plant. It is a plant cell. Plant cells are also eukaryotic. So we're going to talk about eukaryotic cells and prokaryotic cells, but I can tell this is a eukaryotic cell because it has a nucleus, and the DNA resides inside the nucleus. Prokaryotic cells don't have that. You can also see in this plant cell that it has these organelles called chloroplasts, which carry out photosynthesis. It also has mitochondria, which carries out cellular respiration. So basically what a plant cell does is the plant cell uses the chloroplast to absorb sunlight and convert carbon dioxide into sugar or some carbohydrate. So it makes its own food. And that food in the form of some type of sugar or carbohydrate is sent, that food molecule is sent to the mitochondria, and the mitochondria breaks down the sugar. And in the process of breaking down the sugar in the presence of oxygen, it ends up generating ATP for energy. So the mitochondria is the powerhouse of the cell, and it makes the energy that the cell needs. There are other structures in this cell, the rough endoplasmic reticulum, and then outside that, the smooth endoplasmic reticulum. You also see this large central vacuole that stores water for the plant. You see the Golgi apparatus over here. So we're going to talk about cells. There's a little bit of information in the first beginning slides on microscopes. So if we were in lab, if we were doing in-person lab, we use a light microscope, a compound light microscope. And that's what your V-scope is in your science interactive labs. This slide just shows you the size range. So I was just telling you about bacteria. So the smallest bacteria are 100 nanometers, and some bacteria can even be larger than that. So let's just say that bacteria are about because I like using the same units. So let's say a large bacteria is 1 micrometer, okay? And then most plant and animal cells are going to be up to 100 micrometers, between 10 and 100 micrometers. So that's going to be 10 times or 100 times bigger. But your smallest bacteria, which are about 100 nanometers, can be 1,000 times smaller than the cells that are eukaryotic, the plant and animal cells. So this is just showing you different cells and their size compared to other cells. And also it shows you like the size of an atom compared to a cell. Viruses are smaller than bacteria. Some bacteria are the same size as your organelles called mitochondria. Most bacteria are in this range, and then most plant and animal cells in this range. So most of the time, if we go back to our paramecium and our dot, the dot is representing a bacterial cell, which is a prokaryotic cell. And prokaryotic cells are much smaller than eukaryotic cells, like the one that forms this paramecium. So it can be 100 times smaller or 1,000 times smaller, somewhere between 100 and 1,000 times smaller. All right, but we can see prokaryotic and eukaryotic cells under the light microscope. And light microscopes can magnify our specimen up to 1,000 times bigger than its actual size. But you can't normally see organelles except the nucleus. So if we want to see the organelles or if we want to see more detail, we use an electron microscope, either a scanning electron microscope, SEM, or a transmission electron microscope. And I'll talk to you about those in a minute. But I want to show you the images at the top. What we're looking at are different types of staining techniques and different techniques like phase contrast and different contrast techniques. But all of these images at the top are of epithelial cells, human epithelial cells. They look like the cells that we see under the microscope when we try to look at our cheek cells in lab. So I like the phase contrast view because I can see like individual cheek cells. There's one and the nucleus is in the center, and then here's 2, and then here's 3 and 4. We can only see a little bit of the 3rd and the 4th cell over here on the left. But anyway, even with the light microscope, like we're seeing images up here at the top from a light microscope, we can still use different techniques to make the cells appear, to be able to see the cells more clearly, staining and phase and contrast techniques. But if we just need to see a small structure and we need to see more detail and we need to see it larger, then we just need a more powerful microscope, an electron microscope. So the scanning electron microscope is good for looking at the surface of a specimen and getting detail of the surface of the cell. So if you look, the surface of this cell, you can really see that it's covered in these little hairs called cilia, and you can really see that detail. But if you want to see the internal structure of cells, like the organelles, let's say you just want to look at the mitochondria inside of that paramecium, then you would use a transmission electron microscope. So electron microscopes, one thing you cannot do with them is look at living cells. The cells are dead when you view them through an electron microscope. So we use a light microscope so that we can see living cells and structures that we know we can see magnified up to 1,000 times. But then if we need to see something smaller, if we need to see more detail, then we're going to use an electron microscope. Okay. All right, so this is slide 15, and now we're going to look at the notes on the 2 main types of cells. 2 main types of cells are prokaryotic and eukaryotic. There are 3 domains of living things, and you see 2 of the domains listed here. The domain bacteria and the domain archaea. Every member of these 2 domains is unicellular and prokaryotic. So there are differences between them, but they're all unicellular or single-celled organisms, and their cells are prokaryotic. They don't have a nucleus. Then the third domain is not listed, but it's the third domain is the eukarya, which comes from the word eukaryotic. So the third domain of living things are the eukarya, and they consist of the protists, fungi, animals, and plants. Those are the four kingdoms of living things within the domain eukarya: the kingdom protista, the kingdom fungi, the kingdom animalia, and the kingdom plantae. And all of them, their cells are eukaryotic. They do have a nucleus. Similar to the paramecium. The paramecium we looked at in the first slide is a member of the kingdom protista. It's a protist. It's a protozoan. Now, it doesn't matter what type of cell, prokaryotic, eukaryotic, all cells have these components. All cells have a plasma membrane surrounding the cell. All cells have a fluid or semi-fluid substance inside called cytosol. All cells have DNA, and all cells have at least one chromosome, because a bacterial cell tends to just have one circular chromosome, one large circular chromosome. But chromosomes carry genes, which are pieces of DNA that code for a specific trait or specific protein. So the chromosomes carry genes or pieces of DNA that code for proteins. And so those chromosomes carry pieces of DNA that code for traits, and all cells have chromosomes. And all cells have these structures called ribosomes. And ribosomes make proteins. So the DNA works alongside with the ribosomes, works together with ribosomes to make proteins. That's what DNA actually does. Specifically for prokaryotic cells, even though prokaryotic cells have all the structures that was on the previous slide, they don't have a nucleus. If they don't have a nucleus, but they do have DNA, then what we say is that even though the DNA is not enclosed in a nuclear membrane or enclosed inside of a nucleus, it kind of floats around in the cytosol of the prokaryotic cell. And the region that we find the DNA located in is called the nucleoid region. But the DNA is not enclosed in a membrane. There is no nucleus in prokaryotic cells. Prokaryotic cells also have no membrane-bound organelles. So they have ribosomes, because ribosomes are not bound by membrane. But they're not going to have Golgi, they're not going to have endoplasmic reticulum, they're not going to have vesicles, they're not going to have mitochondria or chloroplast if they're plant cells. Prokaryotic cells are also going to have cytoplasm surrounded by plasma membrane, and then there's going to be a cell wall and even a glycocalyx outside the cell wall for extra protection. So this is a typical bacterial, it says bacterial cell here, but then up here it says prokaryotic cells. So I guess it could represent a bacterial cell or a cell of an archaea, because both of them are prokaryotic. So what you're going to see is the DNA is all located in this one single circular chromosome. It's just all folded up inside the cell. But that chromosome is not inside of a nucleus because the bacterial cell doesn't have a nucleus. It's located in the nucleoid region. You see these little dots, individual dots, they are ribosomes. Ribosomes make proteins. The plasma membrane is the inner membrane, and then outside of that there is a cell wall, and outside the cell wall is a structure called the glycocalyx. So the cell wall is for extra protection. The glycocalyx is for protection and attachment. It helps the bacterial cell attach to surfaces and other bacterial cells. And there are also these little structures called fimbriae, which are little fibers, little bristles that will help it attach to other things. And some bacteria can move, so they have flagella that will help them to swim. This says this type of bacteria is called a Corynebacterium diphtheria. And so I'm assuming it causes diphtheria, which is a disease that we get vaccinated for. And you can see the transmission electron micrograph image. This was taken with a transmission electron microscope. You could never see this much detail in a light microscope of a bacterial cell. We see bacterial cells, if we're able to see that it's shaped like it's rod-shaped, then we're lucky. We're either going to see a circle, a rod-shaped structure, or a spiral-shaped structure, but that's about all we can see. We definitely can't see any detail with the light microscope. So we use electron microscopes when we can because they're very expensive, but we use those to look at prokaryotic cells. Now, eukaryotic cells do have their DNA residing in a nucleus. The nucleus has a double membrane outside of it called the nuclear envelope or nuclear membrane. So eukaryotic cells do have a nucleus. The DNA resides in the nucleus. Eukaryotic cells do have membrane-bound organelles like mitochondria. Eukaryotic cells have cytoplasm, and they're generally much larger than prokaryotic cells. So I'm going to talk to you real quick about the surface area to volume ratio of cells. So you may realize, hopefully, that no matter how big they are, cells are still small. So why do cells have to be so small? And it has to do with their surface area compared to their volume. So the surface area of a cell is basically the amount of plasma membrane that covers that cell. So we need a large plasma membrane compared to the volume inside the cell. And you can only get that with small cells. So I'm going to compare to you these two cubes, and I'm going to show you the math, but we don't have to do any math. But these two cubes, the 1 and the 5, the 1 unit and the 5 unit cube, we're going to pretend that they are cells, and the cube that's 1 unit big, I guess you say 1 cubic unit. If it was centimeters, we'd say 1 cubic centimeter. But anyway, the smaller cube, when you calculate its surface area, you basically, surface area is calculated by doing the surface area of all 6 sides. So each side would be 1 unit square because it's 1, the height is 1 and the length is 1. So you go 1 times 1, length times width. 1 times 1 is 1. So the area of each side is 1, and then you multiply that times 6 because there's 6 sides. So you get this total surface area of the small cube is 6 units squared. And then the total volume is length times width times height, which is all 1. So 1 times 1 times 1 is 1. So we say it's 1 unit cubed, or 1 cubic unit is the volume. When we do the surface area to volume ratio, it's 6 to 1, and 6 to 1 equals 6. So that is the surface area to volume ratio of our small cube. If we do the same math steps for our larger cube, the surface area to volume ratio is 1.2. The surface area is 150, the volume inside is 125. So you can see the surface area compared to the volume. That ratio is much smaller. So what we're looking for is cells have to have a large surface area to volume ratio. They have to have a large surface area to volume ratio. And the only way to do that is for them to have a small size. So the question that you might be asked is, why are cells so small? Why can't cells be larger? It is because they need a large surface area to volume ratio. Okay? And then this is an image of a typical animal cell, eukaryotic cell, and you can see the structures. We're going to talk about all these different structures and what they do, but I just want to show you what they look like. So in this typical animal cell, or typical eukaryotic cell, I just know it's not a plant cell because I don't see any chloroplast and it doesn't have a cell wall. So a typical animal cell is going to have a plasma membrane, And then inside the plasma membrane, it's going to have, I don't see it labeled, but the fluid is going to be called cytosol. There's going to be a nucleus, and inside that nucleus, surrounding the nucleus is the nuclear envelope. Inside the nucleus is a structure called a nucleolus, and the nucleolus makes the ribosomes, and we learned that ribosomes make proteins. And then you also have your DNA in a form we call chromatin. They're not condensed and coiled into chromosomes, but it's in a form we call chromatin. So the DNA is always going to be chromatin when the cell is not actively dividing. And then that chromatin will condense when the cell begins to divide, when it begins to undergo mitosis. Outside the nucleus is the rough endoplasmic reticulums, the system of channels with ribosomes attached. Those little dots are ribosomes. And we're going to call it ER from now on. And then the smooth ER is outside of that. It doesn't have ribosomes. You can see at least 3 mitochondria, and they're going to produce ATP for the cell. You can see a vesicle called a peroxisome, which functions in breaking down fatty acids. A vesicle called a lysosome, which functions in breaking down almost any other type of substance. It has digestive enzymes in it. See a Golgi apparatus, which is this green structure that's organized into stacks. It's going to package whatever is produced from the rough ER. It's going to package it into vesicles. And let's see, we also have these protein fibers or filaments called the cytoskeleton. There are 3 different types: microfilaments, intermediate filaments, and microtubules. We see folds in the surface of the plasma membrane called microvilli, and the purpose of microvilli is to increase the surface area even further, to increase the surface area of the plasma membrane even further, maybe for absorbing nutrients. Then we have this particular cell has a flagellum, which is a long whip-like tail, and the centrosome is the area that produces microtubules during cell division. And this centrosome contains these little yellow barrel-shaped structures that are lined up perpendicular to one another. They're called centrioles. Centrioles. They're located in the centrosome. Now, a plant cell doesn't have the centrioles, but it does have the centrosome, which is more like a complex. It's not really a structure. It does have a centrosome, but plant cells don't have these centrioles, these yellow centrioles. So I'm going to stop here. This is slide 24. We'll begin the next recording with slide 25.